community that works on this crop, and also Eve. It's difficult to get a hold of her, and yeah, I feel very privileged that she is with us today and is able to share her vast experience and her reflections on Oka. So thank you again, Eve, for accepting to be part of this seminar for PhD students from so many different countries, and it's and we really look forward to your talk. So please go ahead. Thank you.
Thank you very much, Steph. Thanks for the invitation. Can you hear me okay?
Yes, here in Wageningen we hear you well.
Yes.
Okay, yes. Great. Yes, I think I have too many slides, so I'll watch the time so that I don't go more than a half an hour or so. But yeah, I thought I could talk some about my work over the years on the Andean tuber crop Oca, which is Oxalis tuberosa, and look at I'm glad I picked some of the slides that I did, since Steph was talking about seed networks and seed systems, and I had decided to feature some not very new research, but that does talk about movement of clones of Oca over the landscape, or over larger scale movement that is not my most latest. More of what I've been doing more recently has to do with trying to look at the origins of Oca, its domestication and polyploidy, getting back to my earlier goals. But oh dear, when I'm in these presentations, sometimes it doesn't let me advance to the next slide. Come on. Nope. Do I have to use that thing at the bottom? No. Okay. I might have to get out of. Oh no.
Sometimes you have to click on the screen one time and then put the page down.
Oh yeah, it gives me that thing down there. I don't like using those buttons, but if I have to when I'm in a presentation, I guess I have to. Okay, so Oca, Steph already showed a few pictures of Oca. It is a very diverse crop, not only visually, but nutritionally as well. And a couple of the features that I have been working on that might sometimes make it challenging to study is that it is octoploid with 8 sets of chromosomes, which makes especially some of the DNA-based research challenging. And I'm not going to go into that very much, well, only slightly today, but then the fact that it is vegetatively propagated, so essentially clonal, and that is similar to potato and a number of the other tuber crops that are also primarily vegetatively propagated. So in the Andean region, thousands of years ago, the ancestors of the Quechua and Aymara people today domesticated tuber crops of 4 different plant families. There's not only the potato that's bred around the world, it is now a globally important crop, but plants of 3 other plant families, Oyuco, Mashua, and Oca. And so even though their tubers grow similarly and might look similar, when you see the above ground part of the plant, you can see that they are from 4 completely different plant families. So even though they're tubers, that doesn't mean they're related to potatoes. I have to keep reiterating that because sometimes people keep going back to thinking it's related to potato. It's not. So Oca is one of the other Andean tubers, and other than the potato, they did not spread around the world much yet. But they are very important in the Andean region where they are grown. They are most abundant in southern Peru and in Bolivia, but they are still grown in other places further north and south in the Andes as well. So they have a very important role in the crop rotation system because you can't keep growing potatoes year after year on the same soil without getting a buildup of crops and diseases. So it's necessary to have different crops that can be for part of the crop rotation system at the higher elevation parts of the sorry, I'm realizing that I have to turn on a pointer. The higher elevation areas are usually where the tuber crops are grown. So oca is important in that crop rotation system. And also the diversity of different crops is important for food security in the Andean rural communities. And so another thing, when we think about its importance, one of the questions I have is whether oca might have been, can't prove this, but that it might have been more important in Inca times before colonialism. And a couple of hints that come from early colonial times. One is this Hero vessel, a very large wooden drinking vessel that is called the Caro of Agriculture because it features many different crops around the different sides of the vessel. And one of the featured crops is oca. This is clearly oca with the yellow flowers, the leaflets, the 3 leaflets on each leaf, and then the tubers down here. So oca is one of the featured crops on the Caro of Agriculture. There's the oca, below it, what is probably potato. On this side, a couple of different chili peppers, maybe rocoto and ají, and other crops around the other sides. So it was definitely, this Caro was what archaeologists mean by the transitional period, means that early in colonial times when the Inca craftsmen who were alive during Inca times are still carrying out their crafts as they would have done in Inca times, even after the after the arrival of the Spanish. So this would have been an Inca craftsman who made this wooden Caro. Another place where there are hints is in the Chronicles of Wamampoma. And in this drawing, he depicted potato and oca planting time, with this man using a chaquitaclia and the woman planting some tubers that here might be potato, but oca is definitely mentioned in the title of the drawing. And at one point I had borrowed an edition of Wamampoma that had an index, and I noticed that the number of times that oca or kaya, the dried oca tubers, or other things having to do with oca were mentioned in the index was probably higher than almost any other crop, with the exception of potato. So it made me wonder if oca might have been more important back in Inca times than after the arrival of Europeans. A number of years back, I tried to look into what the archaeological evidence of oca was, and I am not an archaeologist, so I probably missed a lot. But this is what I could find. That Caro is probably this one up at the top, the least old of the evidence. And a number of these things, I think I put the asterisk on the ones that are depictions of oca in artwork, other than remains of the plant itself, but depictions in art. So that Caro would be here, and then there are older ones, but oca is also found in Iterero Cave in Ancash, and this has some of the oldest archaeological remains of agricultural crops in the Western Hemisphere. And oca tubers were found all the way down to the very oldest strata in this cave. And Smith, who was the archaeologist, had mentioned that this is the area around Ancash where Gitero Cave is found. He said, if it could be shown that this is outside the range of the wild relatives, then that would be stronger evidence that the remains that were found in Gitero Cave are actually of domesticated oka. So we'll come back to that a little bit later. I tried to feature more on cultivated oka for this talk, but I have been doing work on the wild relatives and how they are related to the crop. So I'll divert a little bit into that right now, especially with relationship to that question of the archaeologist at Gitero Cave. So this map shows all these, the entire range throughout the Andes from Venezuela down to northwestern Argentina, with a few in Central America, is the members of the lineage, the clade, within the very large genus Oxalis that comprise the relatives of oka, probably at least 36 species. So all of these black dots are plants that are do not have tubers and are mostly diploid. The colored dots represent the places where we have found tuber-bearing wild plants in 4 different regions: Central Peru, in Lima Department, Southern Peru, in Cusco, Apurimac departments, in Bolivia, and also in Northwest Argentina. And some of these have names, Oxalis pichensis in southern Peru, Oxalis chicligastensis in northwestern Argentina, but the ones in Bolivia and in Lima department are not named species. They go from very tiny ones. Oxalis pichensis are mostly barely the size of a pea. The ones in Bolivia, some of them get up as the largest are maybe the size of my little finger. So they don't get as large as the cultivated ones, but the larger ones are these ones in Bolivia. And so I will talk about their relationship to cultivated oka, but I need to introduce another character in the plot. Back when I was a graduate student in the 1990s, I interviewed farmers in the district of Pisac, which is about an hour's drive from Cusco City, asking them about the different kinds of oka that they grew and the different features of the characteristics of the different kinds. And so in this family's oka, there's maybe 1 variety in the back pile and 3 or 4 different kinds in the front pile here, and asking people about how they classify and name the different oka varieties. And one of the most important divisions among the different varieties was that some of the varieties taste very sour and are only used for making kaya. Kaya is oka processed by soaking in water first, and then for maybe 3 or 4 weeks, then exposing them to freezing and drying until they form dried tubers that will then keep indefinitely. And certain varieties, other varieties can be used for kaya, but the very, very sour ones are always used for kaya. And they're usually, here's the some oka soaking in a little pool of water. You can almost see some of the tubers around here, a little bit of bubbling because some fermentation is taking place here. So that's the first stage, is soaking in either still or running water, depending on the region. And so usually people are growing most of their oka in mixtures. And so these top 2 rows would be what they would call waiku varieties. They each have their own names. These are kind of 2 by 2 of different varieties, which each have their own names, but they are classified as basically the ones that are boiled after a few days in the sun to sweeten. So these are grown as a mixture, but I learned that many of the farmers would grow these very sour ones called poshko, grow them separately from the others because they're only going to be used for processing into kaya. Occasionally, they might include these kusipata in there as well because they're not as sour, but also have a more resistant flesh. So I learned that they were growing them separately from all the other okas that are grown together. And so we were paying attention to how those might be different from the majority oka. This is depicting some results from when we used AFLP, a DNA-based method that isn't used as much anymore, but we were focusing on the wild tuber-bearing populations from Peru and Bolivia and Argentina, and comparing that to a sample of cultivated oka, both the white goat varieties and the kaya processing varieties of poshko. And so we can see here that actually the waiku varieties and the poshko or kaya processing varieties were separating into slightly separate little clumps. This is an NMDS analysis, so really what it's depicting is how close things appear on this diagram indicates how similar they are in the particular data that we're using, which was almost 400 AFLP markers. So the ones that were most similar to oka were the wild ones from Bolivia, followed by the ones from Argentina. And it turned out then that the ones from Peru showed more different or distant from oka in these particular markers, which was interesting because my earlier work in my dissertation using Sanger sequencing of 2 different loci, with those markers, we thought that Oxalis pichensis and Oxalis, the ones from wild tuber-bearing ones from Bolivia, might have hybridized to form oka. Well, with these results here, it doesn't look like Oxalis pichensis is actually a progenitor of oka, although it might have had some role to play based on those earlier results. I'll just point out here what we know now about the ploidy levels of these from various different sources. The majority of waiku oka are octoploid. Several of the wild ones are tetraploid. The ones from Lima were hexaploid. With respect to the kaya oka, interestingly, some of those are tetraploid, and I'll come back to that. Also tetraploid, so I'll come back to that shortly. But what these were telling us was that the wild ones from Bolivia and Argentina seem to be more likely to be the progenitors of cultivated oka than the ones in Peru. So if these ones were more likely so far to look like the ancestors of cultivated oka, and the ones in Peru were less likely. If we come back to the question about the Guitarero Cave, the okas found in Guitarero Cave, and if they are outside of the range, here's where Guitarero Cave would be on this map. So it's not only separate from the wild tuber-bearing ones in Peru, but if these ones are not the ancestors of oka, then it is very separated from the ones in Bolivia and Argentina. So that would indicate that, yes, Guitarero Cave is outside of the distribution of the likely progenitors of cultivated oka, or at least where those are found in the present day. We don't know exactly where they were found 10,000 years ago. So coming back to those poshko oka, this is some work by one of my former graduate students, Elizabeth Jane Bradbury. This was quite a while ago. She had her first child while she was a graduate student, and that baby is now in secondary school. So this was a while ago. So she started with a pilot project with help from SEEP to grow a selection of ocas in a common garden situation, including some of SEEP's collections as well as some of my collections from fieldwork. And she found, just in terms of pH, that these Poshko varieties were definitely lower pH, more acid than the majority of the Waiku boiling varieties. And she did a small pilot study with the measuring oxalic acid of a smaller number and said that, well, we had lower pH and higher oxalic acid concentration in the Poshko ocas than in the Waiku ocas. But then later she did a much larger selection of the Poshko ocas and found that some of the ones that were classified as Poshko ocas were octaploid and others were tetraploid. And then comparing that to the oxalic acid concentration, the ones that were octaploid had a lower concentration of oxalic acid, similar to that of the majority octaploid ocas that are Waiku varieties. But some of the Poshko varieties were only tetraploid, but had approximately three times the concentration of oxalic acid in those tubers. So we weren't too surprised to see a higher concentration of oxalic acid, but we were definitely surprised to see that some of them were tetraploid and not all of them. And so this correlation between the ploidy level and the oxalic acid level was a new finding. So we had to slightly modify the conclusion that the lower pH and higher oxalic acid was in the tetraploid Poshko ocas than in the octaploid Poshko or Waiku ocas. And here I just like to give a little plug for ethnobotany, because if I hadn't been surveying and talking to the farmers in that area of Pisac, and learning that they grew their Poshko ocas separately from the other ones and treated them almost as if they were a different crop, we wouldn't have been paying attention to these different results and looking for the differences between Poshko oca and Waiku oca. So this finding probably would not have happened because we wouldn't have been seeing it even if it was in front of our eyes. So talking to the farmers really is important. So that was 20 minutes. I have too many slides here, but I want to say it's convenient because Steph was talking about seed networks and seed systems, and I was interested in this question. So maybe I'll at least do some of these, but I'll try not to run too long. This is a figure from Carl Zimmerer, and if you all are in this Paucartambo in the center of the slide here, this is from work of Carl Zimmerer, and he was surveying farmers about where they were getting their potato seed, whether locally or more distantly, and so in order to try to study the seed flows within and between communities. And so this was done at a particular scale and was done using surveys of both the farmers and market vendors to see how the tuber seed flows were happening. And so I was also interested in this and thought, well, sometimes they travel long distances. Sometimes tubers are put up on a truck and shipped fairly long distances, and maybe most of them get eaten, but a few of them might be planted. So that could be some, in the present day, some very long distance seed flows among different areas. And a number of years back, there were surveys of in markets around Lake Titicaca, and found what they were calling ancient trade routes between major cities in the Andes, even across the international border between Peru and Bolivia. And that made me wonder, well, could that mean that the same clone of oca, or maybe potato, could be found all the way from Cusco, Peru to Cochabamba, Bolivia? How far do these seed flows go today? And what might be patterning where they go and where they don't go? So, had a project in Peru, collecting oca from all along the Andes in Peru. And one of the questions was, to what extent do the languages shared or different languages pattern where oca tubers are exchanged more or less? So this is a map of the indigenous languages. The red is Aymara around Lake Titicaca and further into Bolivia. There's this little dot here is a locally restricted and disjunct from the rest that is here classified as Central Aymara. All the other colors are different variations of Quechua. Quechua in Bolivia isn't too different from Southern Peruvian Quechua, but then there are the difference between Southern Peruvian Quechua and Central Peruvian Quechua is very different, and they barely understand each other, so that linguists now say that we shouldn't even call these different dialects of Quechua. They're actually should be considered different languages in a language family of Quechua because they are mutually unintelligible or barely intelligible between them. And there's certain areas, because of how people have been moved around, where there's a sharp break between the Southern and the Central kind of languages, and then that between the Northern. So I wondered, could there be a pattern of exchange of oca clones that would reflect languages, and also whether it would reflect topography or other patterns. So the field work was done, well, almost 20 years ago, in 2005 and 2006. I was collaborating with the International Potato Center and also with INEA in Peru, the National Institute of Agricultural Research. And most of the geographical genetics or spatial statistics were with the collaboration of Brian Epperson from Michigan State University. So this is showing collection localities around Peru. Note that even some of them that look like they're pretty close together on the dots, because of the topography, it could be hours and hours of travel just between a couple dots that look like they're on top of each other here. I was traveling, the agreement with INEA meant that I was always traveling with one of their coordinators of genetic resources at 5 of their experiment station. This points out the people who were those coordinators of genetic resources that I was traveling with. And there were a lot of other people who traveled with us and helped in the field or in laboratories at SEEP and other areas. Many people to thank for the field work and the laboratory work. So we tried to do random sampling in the field if at all possible. And if the oca had already been collected, because over the course of months, we didn't always get there before the harvest, we would harvest randomly from the oca storage, which might be inside a building or under on the ground outdoors, protected by piles of straw, or up on a platform protected by straw. I only saw this once, but I would basically close my eyes and reach into the pile and pull out tubers one at a time at random to try to get a random sample from storage with the farmer's permission, of course. And even before we got into the laboratory, just looking at the morphological diversity, we could see some places where there was a lot of diversity. This is in the upper left, one family with a whole lot of different kinds of oca in their own collection, and others where there wasn't much. The lower left is a different family's collection where 40 tubers, it looks as if there's only 2 different clones there. So not so much diversity in other places. And just looking at what they looked like from different parts, these rather thin, narrow, white tubers with a lot of little babies coming off of the sides were found really only in these collection localities on the eastern side of the Andes in central Peru, but they were quite predominant in those particular areas. But just those 3 areas, not in other places. These ones were these nearly black ones were distributed a bit further, more widely here. But these kinds were found just in 3 localities in the Aymara speaking area of Peru. And these ones also yellow with red eyes. There are other yellow with red eyes in other areas, but later the DNA kind of distinguished these. These were very predominant in these 4 collection localities in southern Peru where Aymara is spoken, but restricted to that area. These bright orange ones were in 2 localities in Ancash department, but not found in other areas. And here's an example of some tubers that were only found in Cuyo Cuyo on the eastern slopes of the Andes and nowhere else. And actually that turned out to be an awful lot of them we only found in 2 or 3 localities, or even just 1 locality in our collection. So that really has these really restricted distributions of clones has implications for conservation. If you want to collect ex situ, you really have to visit all of these places because there are varieties that are only found in one place. So you won't get them if you don't visit those particular places. Or if you're trying to do in situ conservation, then the one Parque de la Papa in Cusco is not conserving all of these different ones found in different areas of the country. So I just described what we could see by looking at the tubers, but there can be we can be led astray by the color of the tuber because there can be a certain level of somatic mutation, so you can get chimeric tubers and then things that are members of the same clone, but they'll have different colors. So then we started to use AFLP in combination with those morphological data to distinguish among different clones. And in NIA in Peru, the National Agriculture Research Group grew out the plants that had been collected, and they become part of INEA's germplasm collection in their agricultural experiment station that is on these giant Inca period terraces. And so here's the Inca, the Oca germplasm collection. And so grew out tubers from each of 40 different collection localities. Later, we merged some of those, but grew out 30 tubers from each in order for us then to use both morphological characterization and molecular data to distinguish clones. And so these are some collection localities from as I said, some of those 40 localities got merged to form 30 localities if there weren't quite enough different fields sampled in some of the areas. So they were all along the Andes here. And so we had some AFLP work done, Terra Time, as I postdoctoral researcher did the lab work. And as I said, Brian Epperson was the expert in geographical genetics and spatial statistics who helped with the analyses. We were able to distinguish over 200 clones using the AFLP combined with the morphological data. Of those, 3/4 were found in only one locality. That's including all of the clones. The AFLP alleles, none of those were found in one localities. And so for the spatial statistics, Brian said we had to only use those clones that were found with at least we couldn't use the ones that had fewer than 5 repetitions in the whole sample. So out of the 218 clones, we only used 62 for the spatial statistics, and we also excluded AFLP alleles that were less than 5% or more than 95% of the sample. So there needed to be more variation. So Brian used a lot of different R packages and mapped with ArcGIS. And so here I will very quickly try to show you some of these patterns that we observed. So these boxes represent those 62 clones, as long as they had at least 5 repetitions in the whole data set. And each little box represents the distribution in Peru. So if you think of all these collection sites in Peru, but they're squished down a little bit because these boxes are wider than they are tall. So what we saw was 27 of the 62 were only found in one locality of our sampling. Again, a different proportion when we're looking for ones that have 5 repetitions. But even if we're only use those that have 5 repetitions, there are almost half that were found only in one locality in the sampling. So many of them have very restricted distributions. But then I wanted to look at, well, okay, are they restricted at all by languages? So these ones that I circled are ones that are only found in the area of Central Quechua. These ones are only found in the area where Southern Quechua is spoken. And then these ones are found only in localities where Aymara is spoken. And there's only 4 of them showing here, but a couple of these are really predominant. This one, there are 46 repetitions in the whole sample, in this case 30. So a few of these clones were very predominant in these Aymara-speaking localities in the south, and so they represent a lot in that particular area. There were only a few that were found with a longer distribution along the Andes, and I've circled 6 of them here. But I want to point out, because you can't probably see that in the little tiny boxes, that they are usually either on the east side of the Andes or the west side of the Andes and not crossing from one side to the other. And I will illustrate that in a different way here. So, as I said, one of the questions was, to what extent does the topography affect distributions of clones? So here are a couple of examples, some clones that are distributed along the Andes. And we can see that here are some, even though it looks as if there's not too much of a distance between this blue dot and this purple dot, they are not shared across the Andes like this. So if you just draw in the highest area of the Andes, approximately hand-drawn, these purple ones this one clone was found at a much longer distribution than most, found in a lot of different localities, but did not cross over to these other localities on the other side of the Andes, either here or over here. And these ones that were found on the west side were not also found on the east side. That is looking at the high mountains as a barrier. Up here, there is a very deep, low river valley, and unfortunately we didn't have good enough sampling among these different localities to be able to really see whether that formed a really important barrier for exchange of ochre up there. And here again, I'm just illustrating, they're sort of cherry picked, I admit, to illustrate the idea that many of these clones are found in very narrow distributions of maybe 2 or 3 localities, but also that they're rarely crossing among different language, where the geographic areas where different languages are found. So if we're looking at the languages, and there might be a more statistically valid than what I just showed you. Wombling analysis was suggested by Brian Epperson as one way to look at that. And so this is an analysis that finds areas of very steep change in allele frequencies. And so he did that using our Peruvian collections. And there are a couple of places where there's particularly sharp boundaries compared to others. And if you take this graph here and overlay it over Peru, where the latitude and longitude would end up, some of these strong boundary elements are in fact in areas that are edges between different Quechua languages, between Central Quechua and Northern Quechua here, and between Central and Southern Quechua here. And so the wombling analysis does seem to be suggesting that there is an effect of, or a correlation, I should say. I shouldn't give causation here, but a correlation between languages and where there is less exchange of ochre clones between different areas. Okay, maybe I'll skip over a little bit here, or just go really quickly because I know I'm over time. We tried to look at different ways of mapping the thing, and there is this one circuit theory that tries to map population genetics of different animal or plant populations as if it was electricity, instead of kind of more simplistic isolation by distance. And so a program called CircuitScape that tries to look at genetics of populations as if it's electricity. And for this, we needed road data, some of it digital chart of the world, downloaded through DIVA GIS, and it had paved roads called roads, and the unpaved roads were called trails. And then we added additional roads from this cloud-sourced ones, cloud-made. And in other cases, when necessary, we added some from the GPS tracks that I was running all the time when we were in the field. So if a certain road that we traveled was missing from both of these, we added it in from there. So we made these maps of the roads of Peru. Here, the blue is supposed to indicate the paved ones and the red ones as unpaved. I'm not sure why these are all shown as paved up there. I don't think that's correct. But looking at this and then doing the CircuitScape analysis, if we're looking at these 2 localities here and the different roads, you can, in the settings, decide if here the resistance ratio, so pretending that this electricity has a certain ratio of resistance, then on which routes are the ochre perhaps traveling between these 2 villages. And you can model these with different resistant ratios. So what Brian did was analyze this for all of our collections in Peru, and you can see where the more easier circuits and less resistant, less travel here. But what really strikes me about this map is to then compare that to the ancient roads of the Inca Empire. And to me, they're strikingly similar. Maybe that's not too surprising that the highways that we have today travel on the easiest routes through the mountains or along the coast. But it's really strikingly similar to me to see how the results of this analysis look so similar to the ancient Inca roads. So one other thing I'll just point out, I know I'm over, but looking at mapping the elevations in the Andes using STRM digital elevation data from the CGIAR Consortium for Spatial Information. And so looking at the elevations of the Andes, and these are really pretty colors, but this might be a little more useful for our analyses, is that oca is grown in the Andes almost always between 2,800 meters and up to 4,100 meters. I had read that in the past, and in our results, it turned out matched that almost exactly. So in this case, the red is showing the areas that are in the oca zone between those elevations. White is too high, and this pale blue is too low. But then you can kind of see that it would be pretty easy for oca clones to be exchanged from one village to another along this oca zone habitat, where the red is connected from one area to another, and not as easy for people to be exchanging from one side of the Andes to the other. So that matches what we were seeing on the other side. So this was actually work done quite some time ago, but what we intended and still intend to do is to look at that isolation by resistance, the electrical current-like analyses, but include not just the road condition, but also that suitability of the habitat for oca, the elevation that's correct, and that language variation, and see whether, if we model all of these together, whether we get better relationship to the data that we have than simple isolation by distance, which doesn't really work with these data. So I'll close there. I'm sorry I went over. And I hope that these studies are going to provide information that will help to conserve the diversity of oca. I didn't talk so much about the wild relatives this time, but hoping to help provide information so that people in Peru and Bolivia and other parts of the Andes can conserve their diversity for future generations. So I'll end there, and I guess I'll stop sharing, see if there are
No,
questions.
leave the picture on. Leave the picture on. It's such a beautiful landscape, Eve. Now we
Yep.
only see big pictures of human faces, so I wouldn't have minded to see that landscape. So sorry for jumping in. It was a fascinating presentation, Eve, and including all the pictures, of course. And I am not a geneticist, so I look at it with a different perspective. But what I saw you describing is also a fascinating oca genetic, fragmented, isolated, connected landscape. And I was suddenly thinking, last week, we had a presentation from Caio de Sousa about the TRICO trial approach. And I was also very impressed by his presentation about how powerful that methodology approach is to evaluate materials, but maybe also to disseminate materials. And then I thought, couldn't we use it in minor crops? And now I'm seeing this oca landscape and I'm thinking, oh my gosh, what are you going to do if you now nationwide in Peru, or even, Andes-wide, are going to distribute oca materials to do a very serious TRICO trial exercise? in terms of evaluation of crops, that's would be very interesting, I think. But are you then also not, let's say, contaminating some sort of an cultural, historical landscape by throwing in whatever we would like to evaluate? Have you any thoughts about that?
Well, I have the same questions. I have the same questions that you're talking about. I think also what Steph was talking about earlier, when there's an increase in markets in a particular crop, that may be good in some respects, that the farmers are able to sell more, but in the other way, it often does lead to simplification. And if you're going to distribute other clones around the country, that could potentially also lead to more simplification. I worry even like this, the field work was done 20 years ago. And I remember even at the time, in certain areas that we went to, there might be pretty low diversity, except you'd find one or two elderly people who were still growing a great diversity of clones of Oka. And even at that time, okay, that was 20 years ago, I was wondering what's going to happen 5 or 10 years. So by now, are any of those elder conservationists of Oka still alive? And those that are not, did anybody younger take up those Oka varieties and continue them? I don't know that. If we went back to some of those same places, would there be what diversity existed 20 years ago? I really worry about that.
Yeah. Well, again, a question that we can take up again in the course in Uganda. What are you going to do with the potential of some materials versus diversity that might go lost? Good. Sorry for taking the floor, but this was really something I wanted to pick your brain on. Or you want to react to that as well, Steph? Do you have any thought about it?
Well, the only thought I could maybe add is that also we need to re-educate consumers. No, so I think consumers can be an important ally in this, and so a lot of the system from market supply chains, but also how seed systems are regulated, uh, do not value consuming mixtures. No, and so but for consumers specifically, eating mixtures is something really beautiful, and many consumers appreciate consuming mixtures. So and so this whole thing of consuming seasonal, local, but also the surprise effect of mixtures, maybe that can be something that can be combined with keeping diversity in the seed system. But there are very few examples of that. But even for quinoa, there are some quinoa mixtures being marketed, no, with 3 or 4 different varieties, and with one of the custodian groups in Peru for potatoes, their collective brand is called Miski Papa, and it only contains mixtures. So it's kind of the value proposition. Yeah. But let's maybe open the floor for for others, no, but for questions and or comments. Yeah.
Any hands? Any questions? Bernard. Unmute yourself.
Thank you for your presentation. It's quite interesting because that's most.
Speak a bit
Can
louder.
you speak closer to the microphone? Yes, louder.
Okay. Thank you for that interesting presentation because most of my work has been on that line. I'm a rural sociologist, so I've been trying to say that crop diversity is socially structured. And when I looked at, there was a presentation, the first slide on FLP markers at 390, so that Argentina, Bolivia, they seem to be closer. the Peru one seemed far away. So I was just thinking, could it be that there was no social interaction and the Oka couldn't move? Because when you look at it, they are not really far from each other. I looked at Peru, you can see again, they are more or less, they are distinct. Could it be that there's less social interaction among the...
Well, I don't know. I think in that case, I was looking at the wild ones rather than the cultivated ones. So in the later part, looking at the mapping of across Peru only, that was looking at cultivated oca. And so I was looking at where there might be more or less social interaction indeed. In the case of the where the ones from Bolivia and Argentina seemed more similar in their AFLP profiles to cultivated oca, that was more looking at back, maybe 10,000 years ago, which of these might have been either the direct ancestors or contributing their genomes into, if oca has eight sets of chromosomes, I only usually do it like this, then where did all those different genomes come from? Which were when oca was first being domesticated thousands of years ago, are these the wild plants that were brought into cultivation? And so in that case, how much the wild ones move maybe didn't depend as much on human beings until after they were domesticated. So in that case, the question was more having to do with where was oca first brought into the where was oca first brought into cultivation and domesticated. So I don't think we can say much about social interaction 10,000 years ago. So I guess I can't really address that. Sorry.
Okay. Okay. Viriato, you had your hand up or?
Yes. Hi, everyone.
Hi.
First, thank you for the beautiful presentation. I just liked a few things that you say that maybe will serve as food for thoughts for our course. The first one you said was the value of ethnobotany, or the fact that you were able to observe, and I think direct observation or something like that, with farmers for you to give value to I think it's a chemical component in some of the varieties, the OPSA. I'm not sure if I'm mentioning it right.
Oxalic acid.
Yeah. So that was the first important aspect, because I was checking our methodologies. I didn't see much on what can we see in the field from our test book. So maybe that will be the first thing to discuss in the course. The second thing that you illustrated very well was how I believe it's a river, right, that divides the two areas where the diversity was somehow not crossing from one side to the
Well,
other.
I think most of it, for most of Peru, where I drew the white line, that's actually the
Yes.
highest part of the Andes.
Yeah.
I drew another little river, a little line in northern Peru to indicate a river in a very, very deep, low valley. But in that case, I couldn't even illustrate any differences there because we didn't have sufficient sampling of different villages on both sides of the river, unfortunately. But the main one along the length of Peru was basically the highest part of the Andes. And even today, where there are roads that cross the Andes, sometimes there might be only 2 vehicles a day that cross along those. So there's not, even today where there are roads, there's not a lot of interchange in some of those places. But yeah, yeah, the oxalic acid, I think, I actually happen to have at home right now the what Jane was using in the fields. In SEEP had grown them all in their field in order to have them in a common garden, but Jane was using this spear
The
pH
pipette.
meter to poke into the tubers to measure the pH of the tubers, so that we could have a first pass of just how acid are they before going to the lab and looking at the actual oxalic acid or other organic acid. We tried to look at other organic acids because some people had, I'm aware of one study that looked at what happens to the organic acids when people usually leave even the Waiku varieties in the sun for a few days, up to a week, to sweeten in the sun. And in the one study I'm aware of, where they measured, I don't know, 6 different organic acids, some of them just decreased a huge amount. Oxalic acid, which is the one that we're more concerned about because it's an anti-nutritional, it went down quite a bit, some, but the ones that really went down a lot, like down to maybe only 15% of what had been there before, a few days earlier, were some other, maybe malic and tartaric acid, if I remember correctly. I'm not sure, but they may be contributing more to the sour taste. And we might not be worried about those ones nutritionally, but the fact that the farmers recognized that those Poshko ones, I remember when I was asking people to identify clones, and they would maybe say, well, is this, a different one, Senorita or Keliopanti or something, and then they'd take a tiny taste and go, oh no, it's so sour, that's Poshko. So that was something that you can detect the content, a certain amount, something about the content of organic acids just by taste, because it tastes very, very sour, those ones. So they're not as good for eating without the processing.
Okay.
I'm not sure I got to your question, though.
No, they were not questions at all. They were highlights of positive points that you mentioned that can contribute to how we think about our course in the coming week, in the coming weeks. Yeah. So thank you.
Okay.
And I agree with Steph. I think we should try to remove the DUS for varieties in general. The distinction, uniformity, and stability.
That'll be a nice discussion, especially also when Ola joins us, and we'll have a lot of firecrackers in the room. Hey, now I see 2 hands. Romanos, you had your hand up first, and then Fawn.
Yeah, hello. Thank you very much, both of you, for the presentations. It was very interesting, also from a plant breeder's perspective. Inspiring, I would say. I was just thinking about the last conversation that you had, that all these characteristics that we don't want, like the acidity and everything, can be part of a breeding program. But at the same time, I'm afraid also that breeding programs can get rid of these kind of characteristics, but can get rid of also some favorable characteristics, like what you mentioned before with the quinoa and the bitterness and the natural control of the pests. So is there any discussion about how can you instruct the breeders to avoid breeding for the good parts of these crops and leaving back the undesirable characteristics, let's say?
think that maybe even what Steph was illustrating, and possibly it's true of the sour ochas as well, we might not even know what some of those benefits might be in what we think of as undesirable characteristics, bitter quinoa, the saponins in the quinoa, or the organic acids in the oca. We think of them as something undesirable, but it might actually be that they're helping to avoid pests. And Steph mentioned that the birds won't eat the high saponin quinoa. There's some thought that maybe the sour ochas are less attractive to the weevil larvae that are a huge problem in oca, but it's not even certain whether that's true. But if it were, that might be another explanation of why people continue to grow the sour varieties in some parts of Peru, at least, even though they take some more work to process.
Thank you.
Yeah.
Yes, and in general also it maybe goes back to the second seminar, no, from Tricot as well, that breeders, impossible that everyone takes on board all the criteria, no, and so different users of breeding products, or whether they're breeding lines or possible candidate varieties, I think to evaluate together with farmers and to evaluate together with consumers to participatory methods that can help, no? And so, and the other aspect, which is always very difficult for breeders, is to keep diversity in the system, no? Because of course always the tendency of having stable varieties that produce in multiple environments within the country, that always, so that discussion of whether it's possible to release mixtures or whether it's possible to have regional subnational releases depending on the local demand, all of that I think are things that are important to keep in mind for breeders.
Fong, and sorry, it looks like the room is going to be occupied by another group who planned a meeting here, so we need to close down. Sorry for that. Fong, you have a very quick question that allows a quick answer?
I think I just have some point to share, not really questions. Sorry to asking. Yeah, I just asked my colleagues that I can ask. I have 2 points. The first point is I don't agree with Virato that we should remove the DUS, but I think we need to find the appropriate approach for different actors. So for breeders, for policymakers, but also for farmers. But I'm really interested in your 2 presentation, and I also connected to what presented on the last week of seminar. And I think that, yeah, what I learned is actually farmers, they do have interest in growing some local varieties, or I think they existing. And the thing is that the barrier for them that they cannot grow is not mainly root things from they are interested or not, but they are bounded by different conditions that they cannot afford to do that. That's what I learned from one of my previous experiments, that the farmers show very big interest, but to do that is a different thing. So interest, and how you translate from the farmer interest to action that they really can do on the field. So do you think that just targeting the food system drivers is a main approach in the way that how you promote the use of NUS crops? Did you get me clear?
Yes, yeah, very clear, very
Yeah.
clear, and I think it's difficult. So they're both complex questions, no, and I think we should keep them in the pocket for Uganda. But I also think that for certain types of seed systems, the DUS and UPOF and the whole registration procedures are maybe good and justifiable, and for other seed systems not. So I think multiple seed systems can operate in parallel. No, and we don't need to think in black and white. So that's something to keep in mind. And for food systems, as an entry point for promoting neglected underutilized species, I think they are already connected to the food system. No? So it's just about innovation and creativity and how that could help to position neglected underutilized species better. But of course, without consumption, there would not be that diversity. You can't conserve what you don't eat, basically. So they're always connected to the food system. Yeah.
Okay.
Yeah.
Yeah. Okay. Well, it actually is unclear whether another group is coming in or not. It is, I think, a good moment to wrap up. As is obvious, we will continue to discuss all these things next week. As for now, Yves, thank you so much for doing this for us and presenting together with Seb. So
Thank you for the invitation.
Yeah. It would have been wonderful to have you around in Uganda as well, but maybe
I wish I
another
could be there.
workshop in the Andes could also be an
Yeah.
option. So, and Steph, you also, thanks a lot for putting in your time while you were traveling around there. We are going to have one more seminar tomorrow, which is also, ethnobotany and genes and minor crops and stuff. So I hope to see you there tomorrow, same time. Yeah. As for now, have a good day, have a good afternoon and evening, sleep well. See you tomorrow. Okay, bye-bye.
Thank you.
Bye.
Thank you.